Hello everyone and welcome to Instant Biology by Dr. Neelab. Today we are going to continue the lectures in the playlist of Immunology. We would be talking about a very important topic in immunology, and the important topic is precipitation reactions. Okay, so if you remember, in the immunology series I have already posted some of the techniques that are most commonly used in case of immunology. So this is also this is one kind of a reaction basically that is used in multiple techniques in immunology. So what is precipitation? How does it happen? Let us talk about this, and let us also talk about the techniques associated with it. Okay, so let us start. So what is a precipitation reaction, or when you call something as precipitated? So in general terms also you would be knowing that when you say that something is precipitated, you think that something would have been, or something's weight would have increased and it would have settled down to the bottom of the vessel or flask or test tube or whatever you are using. Something would have settled down to the bottom. That is called as precipitation. The weight has been increased and it is settling down to the bottom. Using a centrifuge or not using a centrifuge is a different thing. You can use a centrifuge, you cannot use a centrifuge, but the main point is that because of the increase in weight it will settle down to the bottom. That is called as precipitation. Okay, so what is the main concept, or what is the main thing that is happening inside precipitation? So whenever, if we are talking in terms of immunology, whenever you have a soluble antigen in aqueous solution, we have an aqueous solution, that means we have water, and in the water you have a antigen that is solubilized. You cannot see it. Okay, suppose you take let me just draw it quickly. So suppose this is a test tube. I have water in this test tube and I have antigen in this test tube. Now this antigen is soluble. Soluble means you are not able to see it inside this test tube. Okay, so soluble antigen in aqueous solution, I told you. And now when you are adding antibodies to it, when you are adding antibodies to it, like this, like this, like this, like this, now these antibodies have been added, and because of this what will happen, you already know that antigen antibody reactions are very common, isn't it? So what will happen, the antibody will react with this soluble antigen in the aqueous solution, and what will it do? It will form a lattice. Now what is a lattice? Basically lattice you can consider as a three-dimensional structure which is tangled, intertangled, or it is just mixed in such a way that the weight gets increased. Okay, so what will happen, the antigen antibody will interact in such a way that it will form a lattice, and gradually it, because of the increase in weight, what will happen, it will form the precipitate, and soon it will be precipitated down like this. So you will find a precipitate after some time when you add antibody to this solution. This is called as precipitation reaction. So basically there are three steps. First, antigen antibody reaction, second, lattice formation, third, precipitation. So these are the three steps in case of precipitation. One important thing that you have to remember over here is the antigen is soluble. Okay, you would have understood this. So this we have done. Now the antibodies, the other concept is antibodies that are creating precipitation. So this was the antibody that I have drawn in over here, is in blue color. That antibody is creating precipitation. That is why you will call it as precipitin. This is a specific name given to these antibodies that are causing precipitation, precipitin. Okay, so next important point is that antigen and antibody complex, that means the antigen antibody complex that was forming the formation of the lattice, basically it takes place in minutes. Okay, so in a very short period of time the antigen and antibody react with each other. That is but obvious, you already know about this. So antigen antibody complex reacts with each other in a very short period of time, in the matter of minutes. But the visible precipitate that we are talking about over here, that I have drawn over here, it requires certain amount of time. And what is that time? Around 1 to 2 days. So 24 to 48 hours are required when you would be able to see the visible precipitate in over here. So this was about the initial thing, introduction. Okay, now next important thing is that formation of antigen antibody complex depends upon the valency of the antigen and antibody. What does that mean? The valency. So in a previous lecture I have already told you what valency is. I'm just going to tell you that again. So what is the valency means? If I'm talking about an antigen, so what are the positions or what are the spots or how many spots are there on the antigen which have the ability to bind with the antibody? That is called as the valency of an antigen, isn't it? So suppose this is an epitope. Okay, suppose this is an epitope, this is an antigen, and this antigen has you see multiple points or multiple epitopes to which the antibodies can bind. Okay, so suppose here is one epitope, here is one epitope, here is one epitope. Okay, so next here is another epitope. So on a given antigen there are multiple epitopes. If there are multiple epitopes, then you will say that valence, if there are 1 2 3 4 epitopes, then you will say that the valency of this antigen is 4. Okay, so the number of sites, if it is increasing, number of sites or number of epitopes, if it is increasing, then you will say that valency is also increasing. Okay, more are the number of epitopes, more is the valency. Okay, if I talk about the antibody, okay, so antibody should be bivalent. Understand this, that antibody should be bivalent. It should not be monovalent. That means it should have the ability to bind with 2 epitopes simultaneously. Okay, it should have the ability to bind with 2 epitopes. It should have a true valency. It should have the ability to bind with 2 epitopes rather than being monovalent, that is the ability to bind with only 1 epitope. So that is why I am saying that it should be, antibody should be bivalent in nature. That is, it should have a higher valency, not monovalent. Monovalent means it is having only 1 valency, bivalent means it is having 2 valency. Okay, again the concept remains same, that it is the ability of the antibody to bind with 2 antigens simultaneously. It is called as bivalent. It is called as the valency. If it is able to bind with 2 simultaneous antigens, the valency would be bivalent or 2. If it is having the ability to bind with only 1 antigen, so the valency would be 1 and it would be called as a monovalent antibody. So I believe you have a clear-cut idea about this, the valency of antigen and antibody. So antibody should be bivalent. This is what I have already talked about. And antigen should be at least bivalent, or it should be polyvalent also. This is the example that I had just shown you of a polyvalent antigen, isn't it? So what does bivalent means? Bivalent means 2 copies, at least 2 copies of the same antigen. If I am talking about a bivalent antigen, suppose I am talking about this antigen, this is an antigen. If I say this is bivalent, then it is having two epitopes of the same type, okay. So that is why I've written either it should be bivalent or polyvalent. Monovalent would not work, okay. So two copies of the same epitopes, or it can have different epitopes like the one that I had shown you over here. This should be different epitopes, or minimum two copies of the same epitopes are required. This is the important point, okay. So we have talked about this, the antibody and the antigen thing. Now, how was this ascertained, or how was this found out that we require antigen having at least we at least require a bivalent or a polyvalent antigen, and in case of antibody we require a bivalent antibody? So how was this determined? It was determined by an experiment on myoglobin. So what is a myoglobin? It is a globular protein. You might be knowing that it is responsible for the transportation or the storage, basically not transportation. It is required for the storage of storage of oxygen in case of muscles, okay. Required for the storage of oxygen in the muscles. That is the function of myoglobin. So why am I telling about myoglobin over here? Because this bivalent and polyvalent concept was obtained from this myoglobin experiment only. So when myoglobin was tried to precipitate, when the scientists tried to precipitate myoglobin, it was found that myoglobin had multiple distinct epitopes, but only one copy of each epitope. This is very important thing. So it had multiple distinct epitopes. So I've drawn it in this way. This can be one example of myoglobin, because it is having multiple distinct copies of different epitopes. So this is one epitope. You can see the structure as circular, you can see the structure over here as asterisk, triangle, and half moon shaped structure. So all of them are different in structure. That is what I have written, but only one copy of each epitope is present. You cannot see another circle over here or another asterisk over here. You cannot see that. So this is the important or salient feature of myoglobin. Next, it was observed that it formed the lattice with polyclonal antisera only. Polyclonal antisera means the antibodies or antisera. The antisera means antibodies only. You would be knowing this. So antisera means antibodies. Polyclonal means one that do not have, or one that have specificity against different epitopes of a given antigen, or you can say different antigens also, right? So they do not have monoclonal like functionality. They are polyclonal. They can interact with different antigens also, and they can also interact with different epitopes present on a single antigen. So this is the concept of myoglobin experiment that told us that these are the salient properties responsible when you are looking at a precipitation reaction. I hope you would have clear-cut idea now. Now let us talk about these precipitation reactions in case of or let me just first of all try and draw this. So if I try and draw this myoglobin, so this was the thing. Let me just rub this out. Okay, so if I try and draw this myoglobin precipitation, so I said that polyclonal antibodies would be able to form the precipitation. So if I draw it like this, okay, Now you can see that we have these antibodies. Okay, these are different antibodies. Remember, these are polyclonal. These are different antibodies and they are interacting with different epitopes onto the myoglobin protein. So this was the myoglobin protein, remember? This was the myoglobin protein. It had different types of epitopes like this, like this, like this, like this. So these antibodies are polyclonal, and that means that they are able to interact with different epitopes. So now you can see a lattice is formed. Now this is called as a lattice. You can see that things have intertangled with each other. So this is a lattice, and soon it will precipitate out. But if the same thing is done in case of monoclonal antibody, if you do the same thing in case of monoclonal antibody, then monoclonal antibody would be very, very specific with only one epitope. So this will interact like this. Another antibody would be interacting like this. Like this. Okay. You can see over here that there is no formation of lattice. They are not intertangling with each other. That is why this monoclonal antisera would not be able to precipitate the myoglobin out. So remember, this is myoglobin only. Myoglobin. Okay, I believe you would have a clear idea about this. Now let us move on to the precipitation reactions in fluid. Precipitation reaction in fluid. So in order to understand this diagrammatic representation, let us first start with the axis. So on the y-axis you can see the antibody precipitated, on the x-axis you can see antigen added. So what we are doing is, we have, let us say we have different bottles, and these bottles are of the same type, and in these bottles we have a constant amount of antibody. And on this, inside these tubes, now what we are having, we are adding up increasing concentration of antigen. Okay, so we have a constant concentration of antibody over here, constant concentration of antibody in all these tubes. To these tubes we are adding increased concentration of antigen. That means one tube has lesser, then slightly more, then slightly more, then slightly more. We are adding increased concentration of antigens over here. Okay, then what will you obtain? What will happen? Definitely the antigen and antibody will start interacting with each other in each tube. Antigen and antibody will start interacting with each other in each tube, and you would see, and if we start measuring the precipitate that will be obtained. Now if antigen and antibody are interacting, what will happen? They will precipitate out. What you are doing is, you are measuring this precipitate. So you are just measuring this precipitate. So antibody precipitated, and the amount of antigen that you are adding, you are plotting a graph between the amount of antigen added and the precipitation that you are getting. You would get a curve like this. And this curve is called as precipitation curve. So this is called as precipitation curve. Now you can see, actually you can see these three phases of this curve. In this first phase, you can see the antibody is in excess zone. Why are we calling it antibody excess zone? Because the amount of precipitate over here would not be very high. The amount of precipitate that is formed would not be very high, because understand this, because there is no proper ratio or proportion of the antigen and antibody. Over here antibody is present in a higher concentration, that is why the amount of precipitate is less. You can see the amount of precipitate is less over here. And this plus and minus, what does this mean? So in this antibody access zone, you can see the antibody is in higher concentration, that is why I have written plus 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 plus, and antigen over here, it is in lesser concentration, that is why I have written minus 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 minus. Over here you can see also that antibodies are in higher concentration and antigens, these are red dots, they are present in lesser concentration. Okay, so it's just a curve between the amount of precipitation obtained, amount of precipitate obtained with the antigen added. So it is going like this. Then let us discuss about this zone, about the last zone, or let us just first of all discuss about this central zone. Now you are again increasing the antigen, right? Again you are increasing the antigen. Finally you reach in this region, the middle region, the region that is giving you peak. This is called as equivalence zone. Remember, it is called as equivalence zone. And why we are calling it equivalence zone? Because over here the proportion between the antigen and antibody, it is optimum. And if it is optimum, the highest amount of precipitate would be obtained in this region. Highest amount of precipitate, you can see if you are comparing, if you are drawing the graph, this is the highest amount of precipitate that you are obtaining in this region, okay, called as equivalence zone. I believe you would have understood this. So over here also in this direction, in this diagram, I'm showing that now the antigen is also not in excess, negative, and antibody is also not in excess, so both are negative. Why am I saying negative? Because they are not present in excess. Okay, they are not present in excess. Over here when I was saying that antibody was present in excess, that is why I was writing plus. Antigen was not present in excess, that is why I was writing in minus. Over here both of them are not present in excess, that is why I have written minus over here, okay, in this case. And over here you can see this, the next case, that the antibody and antigen are forming a proper lattice, and because of this lattice there would be a high amount of precipitate that will be formed. Next, finally, antigen access zone. If you just if you have understood these two, you would be clearly able to understand this one. Over here the antigen is in excess, antibody is deficient. So over here also you can see the antigen is present more, antibodies are not present in a higher number. Okay, so that is why in this region also the amount of precipitate that would be obtained would be decreasing in this case. This is the example in case of fluids. This is done in case of fluids. So precipitation reaction in case of fluids, you get a precipitation curve. Next, let us come to precipitation reaction in gels. So what happens when you do this kind of a reaction in case of gels? So what is a gel? Basically you use agar, okay, to create gels. So suppose I had a circular gel, okay, I had a circular gel, and in this circular gel I already have antibodies present in a optimum concentration. So I have already plated in such a way that throughout the gel there is antibody present at an optimum concentration, okay, optimum concentration. So this red dots that I have drawn over here, they are basically symbolizing the antibodies are present over here, okay, antibodies are present over here throughout the gel. What I do is I cut a trough over here in the center of the gel. In the center of the gel, what I do is, suppose this is the gel, in the center of the gel I cut a trough, okay, I remove the gel from here and then I put in my antigen over here, Now what will happen? Definitely the antigen will diffuse towards outwards, okay, it will start diffusing towards outwards, and as it is diffusing, what will happen? The concentration of the antigen will start decreasing as it is moving towards outwards. As it is moving towards the outwards from its source, the concentration will keep on decreasing, and at a particular point what will happen? The concentration of the antigen and the concentration of the antibody will be in proportion, and they will form a precipitin ring. The black one that I'm showing you over here, this is actually the precipitate form. The precipitate is forming a ring. This is called as precipitin ring. Okay, this will be called as precipitin ring. And one more important thing that you need to remember is that area of precipitin ring is directly proportional to the concentration of the antigen. The more is the concentration of the antigen that you have used over here, higher would be the area of the precipitin ring. So this was the area of the precipitin ring. It would be higher, it would be greater. Area of the precipitin ring would be higher when you are using a greater amount of antigen. Okay, greater concentration of antigen. So one thing I forgot, that precipitation reaction in case of gels can be of two types: radial immunodiffusion. So this was the example basically of radial immunodiffusion. Radial, why? Basically it is the antigen is diffusing out in the radius only, right? That is why it is called as radial immunodiffusion. Now another example is double immunodiffusion. So over here it was across a radius, and over here the gel is cast in a basically rectangular fashion, and there are made two troughs, one over here, another one over here, and in these troughs what you do is you put your antigen and antibody. What you are doing is you are putting your antigen and antibody on two different troughs on a rectangular casted gel. So this rectangular casted gel is basically put on a glass plate. So you have a glass plate, you cast a gel around it, you just make two troughs, and then in one trough you place your antigen, in one trough you place your antibody. They start diffusing towards each other, basically not only towards each other but away also, but we are considering the amount that is diffusing towards each other. Now naturally, when the antigen and antibody in a particular area would have optimum concentration, they will start interacting and again will form a precipitin ring. So in this, again there is a precipitation line that you can see over here. So this is again over here, this was in the form of a ring. Over here you can see a straight line that is showing you that precipitation has occurred between antigen and antibody. So these were the two reactions. One was in the fluid, one was in the gel. So I believe you would have a clear-cut idea what is radial immunodiffusion, what is double immunodiffusion, how does the precipitin curve gets formed. So what is precipitation reaction? I believe you would have a clear idea. If you like this lecture, kindly hit the like button and subscribe the channel if possible. So don't forget to press the bell icon. Thank you so much and have a good day.